Hi, Dr. Brian Kaufman, a retired family doctor, CLL patient myself, and I'm the Chief Medical Officer, Executive Vice President, and co-founder of the nonprofit CLL Society. Excited to be back at ASH 2022.
Hello, this is Adrian Wiestner. I'm a hematologist and clinical investigator at the National Institutes of Health in Bethesda, Maryland.
Dr. Wiestner, the treatment paradigm and the success of treatment in CLL has radically changed over the last several years. And the beginning of that revolution was blocking Bruton tyrosine kinase, or BTK, and now we have several drugs that can do that. But it doesn't always work, and the cancer cells can find a way around it. But you and your colleagues at the National Institutes of Health and others have been looking at novel ways of taking out the BTK pathway. Can you explain that to us and why it's so different than the BTK inhibitors?
Yes, so that's very true, right? So the BTK inhibitors now, 10 years in, have really shown a remarkable efficacy across CLL patients and benefited a lot of the patients. But the problem is that in some patients, the CLL cells find a way around the inhibitors. We call that mutations that then prevent the drug from actually hitting its target. And so efforts to then resensitize or to retarget the cell, to find another strategy, have been pursued. So many people will be aware of the noncovalent inhibitors, like pirtobrutinib. They basically do the same in a slightly different way. They inhibit the kinase. They inhibit this molecule that drives the CLL growth. And
a kinase is just an enzyme, right, that speeds up the reactions.
Yeah. Exactly. A kinase is an enzyme, is a switch in cells that triggers a program. And the switch is on when the kinase is active, the program proceeds and the cells grow. When you turn off the switch with an inhibitor, you basically block that signal. Now, when mutations come, then they restore the signal. What's now different with this new way that you referred to is that we actually aim to degrade the whole kinase, so to destroy it.
Degrade means to eat it up, right?
To have the cell, to trigger the cell to eat up that kinase. And maybe if I may use an analogy here, in some ways the kinase is a switch, and the inhibitor turns it off. What you do with the degrader is you basically pull out the whole thing, right? You get rid of the whole thing, you pull out the whole circuit. And that is mechanistically somewhat different, because when we use the inhibitor, you need to have it on, in that case, Bruton's tyrosine kinase, at all times to prevent it from signaling. Once you succeed to actually degrade it, to destroy the whole protein, the cell needs, or cells that would need to resynthesize, to reproduce this whole protein. And we've done measurements of how quickly that a cell can do that, and it takes a cell takes the CLL about 5 days to fully restore those degraded proteins. So we actually get quite a long-lasting effect of degrading this BTK. And what is also different is really, when you use inhibitors, you have a 1-to-1 ratio. One inhibitor molecule can target one kinase molecule. The trick with these degraders is that they only transiently have to bind. So for a short time they can bind to BTK, but then they recruit the machinery within the cells that basically tags, labels, identifies the BTK for degradation, for destruction. And so one molecule of a degrader can go around and hit multiple of these BTK molecules. So that's mechanistically a big difference. And it may explain why at least so far the degrader we worked with, works also against multiple mutant forms of BTK. So different types of inhibitor-resistant BTK can still be destroyed because this interaction may not have to be as long-lasting or as tight to actually do the job.
Well, that's pretty exciting. This is a whole new way of approaching treating CLL. Where is this in development now? And tell us what we know about clinical trials that are results we have, and even if there's open clinical trials patients might be interested in.
Right, so what I talked about so far was pretty much about preclinical work we've done in the laboratory in collaboration with Nurix. And Nurix has developed a molecule that uses this principle called NX-2127, and that's in an ongoing phase 1 clinical trial. And an abstract here at the meeting will be presented summarizing early experiences with this drug, and will be presented on Monday. And I think what the first exciting part is actually it doesn't just work in the lab, it does work in patients. It is still in a time where one needs to figure out how well tolerated it is, but what we've seen is that patients can safely take this drug for extended periods of time. I think some of the patients have been on for more than a year. So as with the inhibitors, it's a relatively long treatment period. There are responses. You have to keep in mind that the patients enrolled in this trial, most of them have seen ibrutinib or acalabrutinib, have seen venetoclax, many of them. Some of them have seen pirtobrutinib. So it's really a group of patients who have gone through all that we now consider standard of care. So it's relatively early to understand really what's the true response rate when you're on it long enough, and how long do the responses last. So I think the main message of the abstract presented on Monday is that this principle can be translated into the clinic. As always, there probably will be a learning curve, how to best do it. But I think it's exciting. This principle opens up a totally new way of actually going after what keeps these cells alive. And you asked about clinical trials. Yes, Nurix has this trial. As far as I know and see on clinicaltrials.gov, BeiGene has a compound that is in clinical, that's registered as a clinical trial at this point. I don't know how far it is, but it's a trial that's up and registered.
So I have to ask, if it takes 5 days to regrow, does that tell us that there might be dosing that could be less often than daily? Is that something that's being looked at in these early trials?
That's a very good question, yes. So it seems that there is a lasting effect even after the drug is cleared, and that may offer an opportunity to maybe do intermittent dosing, so times on and times off the drug. That is being explored. These are really the things you need to work out in a phase 1, right? This is how to best give the drug, how much can you give, yeah.
And the other thing is I've heard that there's degraders in development for other targets, like the BCL2, which is the target for venetoclax. Aren't there degraders in early development there too?
Yes, there are. I'm just not fully up to date what exactly is done, but yes, there are degraders for other targets. There are actually clinical trials in solid tumors with degrader molecules. So the principle of going after a molecule you want to eliminate from the cell is being more widely applied than just to BTK.
So any final thoughts on this class of drugs that you'd want to share with CLL patients?
I think what makes me so hopeful is that whenever we run into a problem with one treatment approach nowadays, it seems like the science is already catching up and giving us the next step. So that was, I think, true when we started seeing BTK inhibitor resistance, that soon after we had the non-covalent inhibitors coming. We have the venetoclax. So it actually seems like the science is keeping up with the pace of the CLL, finding ways around current treatment strategies.
Well, I love your optimism, and we're so dependent on smart researchers coming up with new approaches and to keep us kind of one step ahead of our CLL, which likes to mutate around treatment. So, Dr. Wiestner, thanks so much for all you're doing for CLL patients and your colleagues at the National Institutes of Health. We are so grateful. Thank you.
Thank you very much, Ryan. Thank you.